Good morning, my little mycologists. Today we're going to talk about fungal reproduction and diversity. So fungi are all classified and easily identified by their fruiting bodies, but what's more important is on those fruiting bodies is how they reproduce. So that's how fungus are actually classified, is by their way of sexual or asexual reproduction. So fungus, just like most eukaryotic organisms, have asexual and sexual reproduction. Asexual is super easy. A little structure called a sporangia comes up and then releases spores. So asexual sporangia releases spores. In sexual reproduction, you actually have the hyphae, if you remember, are the structures underneath. Okay, they have basically not male and female, what we call them positive and negative. Okay, because so one hyphae can have kind of like male and female parts. So we designate those as positive and negative. So positive and negative hyphae are going to come together, okay, and then make what we call a zygote, and then that's going to grow up into an actual fungus, okay, and then that'll release spores. So that's sexual, which we'll get to in a second. To the actual life cycle is a little bit more complicated than that. There are 4 different main divisions of fungi. The first one is zygomycota because it makes a zygospore. Basidiomycota because they have basidia, which is their reproductive structure. Ascomycota have asci. And then deuteromycota are the weird ones that only reproduce asexually. So they're named for their reproductive structure because that's how they're classified. Okay, so sexual reproduction in all fungi, except for the deuteromycota, they follow the same general process. This is the process that you need to know, okay? So their hyphae are haploid. If you remember what haploid means, that means that they have one copy of the DNA. So 2 haploid hyphae are going to come together, okay, and they're going to merge, okay? And so that is called plasmogamy, okay, like polygamy. But plasmogamy is when the cytoplasms of the hyphae come together, and that's going to allow the 2 nuclei to be in the same vicinity. Then those 2 nuclei, which are haploid and haploid, so N and N, are going to fuse under karyogamy. Okay, so the karyo means nucleus. The nuclei are going to come together to make a diploid organism. Okay, that diploid organism is called the zygote, just like in us. When sperm come together, you make a zygote, a diploid zygote. Now, this is the only thing that's kind of different from animals, is zygotes, okay, are going to undergo meiosis to get haploid. Remember, we want to get back to our haploid hyphae, okay? And to do that, they're going to make spores, okay? So here is your general life cycle. So you have the mycelium, which is a bunch of hyphae. They are going to germinate into a fruiting body, and that fruiting body is usually a sporangia, which is going to make spores. That's asexual. Okay, and sexual, you're going to have 2 of these hyphae, okay, come together, and the mating types are going to be a positive and negative. They have to come together, and that's called plasmogamy. Okay, plasmogamy is when the cytoplasm joins, making the nuclei together, so they're no longer separated by that septa. Okay, and so what you're going to get is what we call a dikaryotic cell. So dikaryotic 2 nuclei cell. So then karyogamy is going to happen, where the 2 nuclei fuse to make a zygote. Okay, the zygote is now diploid, so 2N, has 2 copies of the DNA. It's going to undergo meiosis, which is going to half the DNA to make spores, and then spores are going to germinate into the actual fungus. Okay, so here's another diagram. Show me. So what happens is, let's start with the spores. So you have, well, we can start with the hyphae. The hyphae are haploid, okay? They're going to come together and fuse to make a diploid, okay? That diploid cell is called dikaryotic because they're 2 cells. They're one cell with 2 nuclei. Then the nuclei are going to fuse under karyogamy, and then they're going to make a zygote. The zygote's going to go into a spore, germinate, and then the cycle starts. Okay, if you have questions, ask me, or you take your quiz, but we're going to have a Zoom tomorrow to talk about it more in detail. So we're going to go through the 4 divisions of fungi. Okay, the first one is zygomycota, okay, or you can say zygomycota, either one. These are named because they are literally a zygote fungus. If you don't know by now, mycology is the study of fungi. So myco means fungus, so literally it's a zygote fungus. Really it's named for the zygospore, so you can see right here, when these fungi sexually reproduce, they make this huge structure kind of like an endospore in bacteria called a zygospore. So they're super easy to find underneath the microscope. These are found commonly in soil and dung, and the most famous example that you need to know is bread mold. So anytime you have mold on your bread at home, that's a zygomycota. Okay, so something that you need to know about them is they are non-septate, which means if you remember from last section, they do not have that wall in between their hyphae. So they have a hyphae, but they don't have the actual septa. Okay, now these are structures you do need to know. So if you look at a typical bread mold or a typical zygomycota, they have 3 different structures. Okay, so stolons are going to go across the bread and spread out. So they're the more horizontal roots. Okay, rhizoids are the ones that go down and anchor. Okay, and then you have the sporangia, which produce the spores. Okay, so you have stolons, rhizoids, and then your sporangia, which makes spores. Okay. They can go through asexual and sexual reproduction. Asexual is very, very simple. The sporangia releases spores. It falls down. That's going to germinate into rhizoids, and then stolons are going to extend out, and then you're going to have your sporangia and then spores. Okay, so super easy asexual. Sexual is a little bit more difficult, but not really because you've got the general structure previously. So you have the 2 different main types of the hyphae, positive and negative. They're going to come together, and you don't need to really know all this stuff. The big thing is the zygote turns into a zygospore. Now, the zygospore is dormant, kind of like a bacteria endospore. So they're going to wait until those moisture environments happen. Okay, so that's why you don't see bread mold all the time. It's usually when you take it in and out of the refrigerator, or the humidity goes up in your house, and then those spores are going to germinate into your actual bread mold. So that's why you don't get mold a lot in your house. It usually has to have a big humidity change, and here in the South, that's a big deal for us. Okay, so this is just a more detailed version, so you can see these are your sporangia and your spores. Okay, you have a rhizoid running across, a stolon running across, and then you have your rhizoids. Okay, so asexual sporangia makes spores. Sexual, you're going to make a zygospore, which is going to turn into a whole entire structure, and they'll germinate, and you can either do asexual or sexual. Okay, division, basidiomycota. These are your cap or club fungus. Okay, I always think basidio is like baseball, so you have a cap or a club. That's just how I've always remembered it. These are what you guys think of your actual fungi as your mushrooms. Okay, so this is the most diverse group. So these are, like I just said, mushrooms, your puffballs, your toadstools, corn smut, which is a fierce shucked row corn. It's like this dark brown fungus that lives on corn. And then rust, that's a completely different thing than on metal. Kind of looks like a smut. These usually have the characteristic of they like to reproduce sexually instead of asexually. The structures you need to know on those are the parts of the microscope. The mushrooms. You have the stalk, which is the stipe. Okay, so think stipe, stalk. Okay, you have the cap. Now the cap is the most important part because it holds all the reproductive structures. So the mushroom cap that you actually eat has gills in it, and that's usually the part that you guys scrape out. In the gills, you have something called basidia. So in between all the gills are a structure called basidia. That's called basidiomycota. Those are what's going to produce the actual spores. Okay, so you have the stipe, the cap, the gills are underneath, and then the basidia make the spores. Okay, so super easy life cycle. Okay, you have the mycelium, which have the positive and negative hyphae. They're going to come together. Okay, they're going to make their fruiting body. Okay, the fruiting body is the mushroom. Inside the mushroom cap are the basidia. The basidia make spores. Spores fall down, and then they germinate. So that's why you usually don't see mushrooms until after it rains, because the spores need moisture. So be on the lookout for that for your extra credit. If you have no idea what I'm talking about, you didn't check the Google Hangout yesterday. The third division is division Ascomycota, just to rearrange the letters and get sac fungi. Okay, and so that's not because the fruiting body looks like a sac. It's because of their reproductive structures are stored in sacs. So this is like your penicillium. This is called a marule or a morale. Those are usually used in cooking. These are your truffles. The really expensive mushrooms that are like $100 for one. If you've ever heard of truffle oil, if you watch the Food Network. These are called your sac fungi. So your yeast are in here that you guys use for bread, but I'll go into all those. And the fungus and lichen. Lichen is that blue green on the pine trees. We'll talk about that in the next division or the next section. Blues, if you're talking like a shower. The thing with him, the Ascomycota, is that they have reproductive structures called asci. Okay, asci are they're called Ascomycota. They're just little sacs that contain usually 8. Okay, of these reproductive spores. Okay, if they're asexual, the hyphae are going to call really, really smaller spores called conidia. Okay, so Ascomycota produce asci. Okay, Basidiomycota, basidia. Okay, Zygomycota, zygospore. Y'all kind of getting the theme here. Okay, some examples of these are morels and truffles. Like I told you, these are your edible ones. Okay, these truffles are snorted out by pigs because they grow at the base of trees usually, and they can go for like $100 for like a single little like mushroom. So these are like delicacies. There's like white truffle oil and like black truffle oil if you're into culinary stuff. Super good, just super expensive. Yeast, we all know that because it's used for baking and brewing, and then we like penicillium because it gives us our antibiotic and it flavors our cheeses. So if you like blue cheese or you've been to France and actually ate like real cheese, that tartness comes from an actual fungus. The last one, which we'll get into more in a couple sections, is division Deuteromycota. The big thing with them is they usually don't produce sexually; they only produce asexually. Okay, these are also known as fungi imperfecti or imperfect fungus because these are kind of like the group that we don't know how to classify them, so we kind of just throw them all in one group. They're very, very close to Ascomycota, but the big thing, their big characteristic also is asexual reproduction, and they're mostly pathogenic. So when you think of a fungus infection, they're usually a Deuteromycota. Okay, so that's like your ringworm, your athlete's foot, and any kind of yeast infection. Okay, tomorrow we'll concentrate on the Zoom. I just want to check in with you guys and see how you're doing, and I'll post the link to that probably today. Hope you have a good Thursday. If you have questions, ask, okay, on the Zoom tomorrow, or you can do it on the Google Hangout. Y'all been pretty good about that. Have a lovely day.